So next we have Dr. Allen joining us today. So she, as it's printed in her bio, is assistant professor of statistics, but she recently got a promotion, so she is now the associate professor of statistics here at Rice University. And she is also at Baylor College of Medicine, where she holds a joint appointment. So her research focuses on developing statistical methods to help scientists make sense of their big data, which is especially applicable in today's world, as well as for us here in Houston with the Med Center just right across the street. So please join me in welcoming Dr. Allen this morning.
Thanks. Thanks very much for having me here. Is my mic on? We're good? Okay, cool. So I am a data scientist, and I want to talk to you a little bit about what data science is, what type of data is out there, and specifically some of the really cool research that you can do in data science. So you probably have all heard buzz about big data, right? How many of you hear big data in your workplace and yes, big data is supposedly everywhere. Basically in every domain, we're able to capture using sensors and other technologies, massive amounts of data. This is true in healthcare, biomedical technologies, internet companies and social media, sensing technology, manufacturing are capturing more and more data. And this is actually an infographic of a projection. They say about 90% of the world's data has been created in the last 2 years. Think about that. And they're projecting that this rate of growth of data is going to stay the same through 2020 and perhaps further. So you can see here, this is where we are in terms of the amount of data, and this is the projected amount of data that we're going to have in the next 4 to 5 years. So this is a massive amount of data. And what I do is I am a data scientist, and data scientists try to make sense of this massive amount of data. And so the definition that I like, everybody has a different definition of what exactly data science is and who's a data scientist. So if you ask 10 of us, we're all going to have 10 different answers. But I really like this definition. I call it the art and science of extracting meaningful and actionable information from data. Okay? And why do I say meaningful and actionable? It's really because when we have these massive amounts of data, there's lots of things in the data that look interesting, right? But what is really truly there? What is there with a high degree of certainty? Can we say this is something that's actually meaningful in the data and that we can act upon? And this is important, for example, in what I do with biomedical technologies, because the action that we're going to, whoa, okay, that somehow went down. And it's back up, excellent. Because the action that we're going to take is going to have a clinical impact on patients' lives, right? So we need to make sure that what we found in this massive slew of data is meaningful and we can make sense of this. So what is data science exactly, okay? So there's kind of a process about data science, and someone has some type of idea, and they're like, hey, we want to find, so in my world, I'm going to talk today a little bit about some of my work in cancer genetics. And someone might say, we want to find a new possible drug target for a particular type of cancer as a personalized medicine therapy. Okay, so that's the idea. You've got to have data that you can look at and mine to answer this question and manage the data and data analysis and communicating the results. And what this really entails is data science, I really want to emphasize, is massively interdisciplinary, and it's really a team effort, right? We have domain expertise, and we have data scientists who specialize in computer science, statistics, and applied math, working together to answer these types of questions. So what I tend to do, I am trained as a statistician, but I'm also here in the electrical engineering department of computer science. But what I do is the data analysis side. And this is, again, trying to find that meaning, trying to analyze the data and mine it to find something interesting. And I break down, if you're not sure what data analysis is, I break it down into, say, prediction and data-driven discoveries. Prediction is pretty easy to understand. How many of you have heard of these driverless cars now that Uber is driving around the streets of Pittsburgh, right? Or Google are driving all around Mountain View and the rest of California, apparently, right? So all of these driverless cars, they're based upon machine learning and data science technologies where they've captured tons and tons of data from images as this car drives along, and the car needs to be able to make a real-time prediction when it approaches something that looks like a stop sign. It needs to recognize that it's a stop sign, make a prediction, this is a stop sign, and act accordingly and stop, right? And it has to do this even with there's some mud splashed on the stop sign or something else. Like, this is really important. So prediction is a very critical aspect of data science and data mining. But what I'm going to talk about today is what I call data-driven discoveries. And that's just taking a mountain of data and finding something interesting in that data that we can act upon further. Okay, so this is less. Prediction is really clear what the goal is, right? We need to recognize the stop sign. That's a very clear goal. Data-driven discovery is a lot more nebulous. So I'm going to talk a little bit about that and about some of my work in this. So when people ask what I do, I say I build tools for data-driven discovery, and I do lots of work in neuroscience and genetics specifically. There's tons of data in those domains. I'm going to talk a little bit about some of my work in cancer genetics today. So the idea is, and a domain I work in, is something I call diverse data. And our goal here is to do some type of data integration. So there's a lot of areas where we capture many different aspects or many different types of data. Imagine social media, right? And imagine all of the data that internet companies are collecting from you. Your Google search history, all of your posts on social media, and I don't mean to scare you, but I mean lots of things, right? They're capturing all of this data. The same thing happens in healthcare, right? We have patients' visits to hospitals and interactions with the healthcare system. We have any medical imaging or tests that have been run on patients, and so forth. This is what I mean by diverse data. There's lots of different types of data that have been collected on the same people. So statistically, I call this problem a data integration problem because we have multiple different types of data that we're trying to bring together and jointly analyze. So specifically, we call this data mixed multimodal data, okay? The idea is for the same set of subjects, right here, patients. So in the example I'm going to talk about healthcare type stuff. So these are patients that have cancer here. We have multiple different types of data that have been collected on these same subjects. Our goal is how do we put all of this data together? And that's what statistical data integration does. It uses probabilistic models to jointly model all of these different types of data together. And by jointly modeling this data, we can make these types of discoveries that move forward things like cancer research and lots of other areas. So the data set that motivates this statistical data integration problem is something called the Cancer Genome Atlas. And this was a massive NIH-funded project that looked at 11,000 patients who had various types of cancers. Their tumors were resected during surgery. And on these tumor samples, they ran every single possible available genomic profiling technology. And these technologies measure different aspects of what's going on at the molecular level of these tumors, okay? And so there's at least 7 types of genetics data available for all of these 11,000 patients, and in total it's 2.5 petabytes worth of data, okay? 2.5 petabytes worth of data. Just as a reference of how much 2.5 petabytes worth of data is, that's about 5 billion pictures on Facebook that have been uploaded to Facebook. 5 billion, okay? So that's a lot of data, okay? That's an awful lot of data from the Cancer Genome Atlas. And the types of data that have been collected are just all these different types of genetics data that basically have, I explain this as they're capturing different snapshots of what's going on in the tumor, but each type of data only takes a picture from one angle. Okay, so imagine if you're trying to figure out what this is and you've got multiple angles, but you need to actually put all the data together to really understand what's going on at a personal level in every person's individual tumor. Just to zoom in a little bit on this and the types of data that are available and why these are important. So gene expression is actually measuring how much is a gene turned off or on. So in every cell in our body, every cell has approximately the same DNA sequence, but what makes a liver cell different from, say, a neuron in the brain is which genes are turned off and on. Okay, so that makes cell function differently. So you can imagine tumors behave really differently from regular cells, and we need to understand what's making these tumors act like tumors, right? And grow and become malignant. So we need to study what are the different genes that are turned off or on that make this tumor cell a tumor cell, okay? And what we're trying to study here, here's two other snapshots of genetic data. So gene expression data measures for every gene in our body, and in our DNA. It measures a measure of how much is turned off or on. And microRNA expression and DNA methylation are a type of epigenetics data. And this is data outside the coding region of the genes that affect whether genes are turned off or on. The reason this is so important and so exciting from a medical perspective is because epigenetics, our genes are kind of fixed. Our DNA sequence is fixed over our lifetime. But epigenetics can change, and they do change according to your environment and lifestyle factors. For example, this is how if you smoke, we know that that is associated with much higher chances of getting lung cancer. It's the epigenetics that are changing in your system. So these are really important to study in cancer. The other reason they're super important to study, sometimes it's really hard if you're developing drug targets, personalized drug targets for cancer. Sometimes it's really hard to turn off a specific gene, but we can develop drugs that target these epigenetic factors that then will turn off a gene that is perhaps causing a tumor growth, okay? So we have all these snapshots of different data. The challenge from a data science perspective or a statistics perspective is that each of these snapshots of data, first of all, they're big. They're on the order of hundreds of thousands of variables that are measured for every single patient. So the data is quite large, but also the data looks very different. MicroRNA expression is this nice bell-shaped curve. If you remember back from your, how many of you had stat classes at Rice back in the day? Oh, it happens, yeah, sorry. Actually, I hear from most people that, they're like, oh, statistics was my worst class I took in college. So anyway, hope that wasn't the case at Rice. But anyway, so back from your statistics class, this looks approximately like a Gaussian or normal distribution, nice and bell-shaped. But this is a variable that's measured between 0 and 1. And this variable here is a count-valued variable that's highly skewed. So imagine when you have all of this data, but all of the data looks very differently, right? We know how to analyze data that looks like this by itself, and data that looks like this by itself and looks like this by itself. But how on earth do we put it together? And this is the fundamental statistical question that I'm trying to answer with my research here. How we want to put all this together is to model everything that goes on in a tumor cell as a massive network, okay? So what you're looking at here, it looks like a massive hairball, right, of network edges. But this is actually a model of all of the genomic interactions that are happening in a glioblastoma tumor cell, okay? And it's color-coded by the different types of interactions between different types of genetic markers. Okay, so, and this has over 1 million edges here, so over 1 million interactions in this cell. So this is what we're trying to model to understand cancer better. So there's been a long history of modeling genetics data using networks, and particularly we go from data that's in a form of a matrix like this, and we use statistical methods to estimate a network of how these genes interact. So each one of these are what we call nodes in a network. Each one of these is an individual gene, and we use statistics and machine learning to understand that this gene is basically talking to this gene. You can look at these links as these genes are somehow connected or communicate with each other right here. Okay? And the reason scientists love these networks and have used these a lot is because it saves them a ton of time. Okay? So how many of you have spent some time in a lab here when you were at Rex? Okay, yeah, so raise your hand. So all that pipetting that you did, right? It's a lot of time and effort. This is strange. Hope it comes back. Anyway, maybe someone else will come and fix it. So you spend a lot of time in a lab, and these networks are really important because they help to generate hypotheses for scientists. So previously, scientists, before this big data era, scientists were having to use their known biology or hunches about what experiments to perform next to test how genes talk to each other and how we can develop drugs for that target specific genes. But now using big data, we basically tell them, if you want to turn off this gene, you need to target this gene. We use these networks to go and tell the scientists, these are the next set of hypotheses, the next set of things in the lab that you have to test. So scientists love this stuff because it saves them a ton of time and money and time spent in the lab. So our objectives here are to use network-type models to develop new machinery that we can use for statistical data integration that's going to be appropriate for this cancer genomics data. So what is out there in terms of these network models? Okay, so there's some models out there that are appropriate for continuous value data, and these are Gaussian graphical models. So we can model data that looks like that bell-shaped curve pretty well. Okay, this is known in statistics. There's also models from the physics literature called Ising or Ising models that we can use to model binary data. So these are yes and no data, okay? And this for genomics is either you have this mutation or you don't have this mutation. But up until a couple years ago, these were the only two known graphical models out there. So how on earth do we model count value data, skewed data, bounded data, and so forth? This is really important because 3 of the major types of genetics data we get on tumors, including the gene expression data of whether genes turned off and on, are actually skewed or count value data. So how on earth do we model these using statistics? So a collaborative team in my lab developed a framework of graphical models via exponential families. So the key assumption here is that every single variable conditional on the others follows an exponential family. What are exponential families? They're basically all those distributions in your stat classes that you took that you probably remember, like Poisson and negative binomial, Gaussian, Bernoulli, etc. All of those are lovely exponential families. So my question is, if we had a count value, a nice little quiz, right? And that's just what you wanted on a Saturday morning. But if you have a count valued random variable, what distribution might you use to model a count valued random variable? Anybody? I heard lots of things. Like pinpoint one thing. Okay, yeah, we might want to use a Poisson. I'm gonna take this. Yeah, so Poisson and negative binomial, if the counts are large enough, maybe Gaussian, right? Like there's lots of different options for modeling these. But the point is, if you have one variable, you know what distribution to use, right? So the idea here is to leverage what we know about these univariate random variables to a multivariate sense. And there's a lot of math involved, but if you make some key assumptions, you can show that a joint network distribution exists for all of these types of data. So this is really exciting because now, not only do we have these 2 types of graphical models, but we can model count valued data and bounded data and so forth. And this is really important because all the data in the world is not just beautiful and bell-shaped, right? There's lots of different types of data. And this allows us to model, this is an example from lung cancer, where we have modeled a gene expression network. So this is, again, genes that are turned off and on and essentially talking to each other in the network. And this is modeled using Poisson graphical models because these are count-valued random variables. Okay? So this allows us to do this. But of course, this doesn't allow us to put things together. So now we can model model a network of how gene expression talks to each other, how the microRNAs talk to each other, and how methylation talks to each other. But what we really want to know is this in between, right? How do epigenetics affect the genes that are turned on? Again, the goal is to identify novel drug targets for cancer. So our framework here is called mixed chain graphical models, and I'm not going to go into too many assumptions here. There's a lot of technical math details. I skipped this for you. It is Saturday morning, okay? So, but there's a lot of math that goes in here, but basically if you make some key assumptions, you can assume that every variable, conditional on the other variables, follows their own different distribution. So me, conditional on you guys, I could be Poisson valued. You could be, say, Gaussian valued, conditional on everybody else, and so forth. And this allows us to model data of all different types in the same joint model. And after a lot of math, you can show that this joint distribution exists, and we have a joint model that we can work on with all data together. These are kind of a picture of what these models look like. So now we can have chunks of data that are, say, continuous valued and binary valued, but also count valued and bounded, all in the same model, and we can study directed influences and interactions between these genes using these somewhat crazy models. And also we can mix them up all over the place. So these are a really flexible class of models that is going to allow us to do really cool things. So if we go back to this, I want to show you a little bit of an example here. But first, the implication of this, so yes, there's an awful lot of math that goes into this, but this is the first multivariate distribution that is appropriate for mixed types of data, that allows you to directly model mixed types of data jointly in the same model. And so this is, we think this is a really big deal, and so this has a lot of interesting implications. So let's look at a quick example of how we're using these types of models to understand big data in cancer, and specifically, again, our goal is to develop drug targets. So we took the Cancer Genome Atlas data, and on my team I have an ovarian cancer oncologist whom I work with, and he was very interested in studying how this is DNA methylation and microRNAs influence gene expression. So again, he wants to know what epigenetic factors are turning off or on bad genes, basically, in ovarian cancer tumors. Okay, there are several genes that are known to be very bad oncogenes in ovarian cancer, and he wants to know, is it possible to find epigenetic factors that can target these really bad genes? That was the specific hypothesis. So we set up a model that looks like this. This is kind of a representation of the type of model that we set up here. And we did a kind of some aggressive filtering to about 300 patients. We had a lot of exclusion criteria that we considered in this particular study here. And so pay attention here. Just remember that blue and green are epigenetic factors, and red are some genes, okay? And this is an estimate of all of these interactions that we have in these ovarian cancer tumor cells. So you see here, each one of these red dots is a specific gene of interest. We filtered these genes, so every single one of these genes is some type of known oncogene that's associated with ovarian cancer tumor growth or is important for survival for ovarian cancer, okay? So every one of these is red dots is a gene of interest, and then we see these blue dots are microRNAs. MicroRNAs tend to turn off genes, and these green dots are DNA methylation sites that can either turn off or on these genes. And notice here, there's a lot of red dots here, but we've got some really big microRNAs here that, I should say the size of these nodes reflects how many connections it has to other genes. Okay, so you see here, there definitely seem to be some really big players that are influencing lots of genes and potentially turning, microRNAs tend to turn off genes, so these guys are potentially turning off really bad genes in ovarian cancer. And so this got our collaborator, our oncology collaborator, very excited. And I'm going to show you one. So you can imagine from this network, we actually found tons of things. He's now running a ton of experiments in his lab to test a lot of these hypotheses. I'm just going to show you one of them here. And this is for microRNA 425. And this right here is basically the neighborhood of influence. These are our predicted, what our model predicts microRNA 425 is influencing here. And look at all of those genes that microRNA 425 is potentially turning off. And specifically, the genes here that are boxed, of the top 10 genes that are known to be highly mutated and potentially causative for ovarian cancer, microRNA 425 tends to turn off 6 out of the top 10 mutated genes in ovarian cancer. So we are really excited about this, right? This is potentially a big finding here, and there's lots of other things of interest. So what my collaborators are doing, so the way that I'm using kind of these machine learning and statistical tools is I built these networks like this, where I make predictions about what epigenetic factors are important. And then my oncology collaborator goes back and he actually has tested these, and he tested 18 of the genes that were predicted here, and 16 of them turned out to be true in the lab, and he's pursuing them further as potential drug targets. So this is kind of a story of how data science can lead to discoveries in medicine. And future work, again, there's tons of applications of this. I use this in medicine because this is, I find the data super interesting, and this is where I work, and there's lots of challenging problems there. But again, diverse data is in nearly every domain. There's so many applications of these types of tools. It's a really exciting area to work in right now. And we're developing all sorts of other tools in my group that are not just networks. Things like clustering and feature extraction and lots of other types of tools to make discoveries from this diverse data. If anybody, I don't know, does anybody an R user in here? Does anybody even know what I'm talking about with R? Oh, we got 2 R users, fantastic. Okay, sorry. R is the statistical computing language, okay? So it's open source, R is fabulous, right? And we do have 2 R packages. If anybody's interested in actually using the tools we develop, the way we disseminate our tools as data scientists is we write software packages that we then release and people can use. And you have some of this. And again, I just want to emphasize this is a really big team project. So I had at Baylor College of Medicine, I had a bioinformatician, a geneticist, and our ovarian cancer oncologist here. So, and then my student at Rice, who did a bulk of, she coded up all of this stuff and ran this and developed the software. It's a big job. And I also had, these are 2 theoretical computer science collaborators as well that worked on a lot of the theoretical and computational aspects of the model. So again, it takes a really big team to pull off a project like this, and I just want to emphasize that. And thank you very much, and I'd be happy to take some questions. Do I have time for questions? Okay, yeah, sure.
I sent off my sample to 23andMe, and they did a test to see what my ancestry is, but they also checked for a whole bunch of diseases. Isn't that a huge warehouse of data?
Yeah, so I'm going to repeat the question a little bit. So the question is about 23andMe, and it's a huge warehouse of data. It absolutely is, and I think they are using a lot of the data to mine and understand diseases. A lot of companies have actually gotten into this game. There's more and more genomic data that is actually publicly available. So the issue is not that there's not tons of data out there on genetics. And again, we've got data on cancer, on neurological diseases. I mean, you name it, there's probably data out there, genetics data like this out there. The issue really is people, data scientists that can come and analyze the data and find something that's interesting, right? We're at the stage where we've collected tons of genetics data, but we haven't yet made sense of all that data and made discoveries. So I think that's the real crux of the issue. Yeah. Question, yes?
You said that of the 18 microRNAs you identified, 16 turned out to be valuable. What does that say about the other 2? Is there something about your model that wasn't right, or something about the data that fed it, or was there something just weird about those that wasn't recognized?
Yeah, so in big data and in what I do, I always tell scientists that my job is to sort through massive amounts of data to generate hypotheses for them. when we generate hypotheses, not all your hypotheses are going to be correct, right? This is why we have the scientific method, right? So, and specifically, when you're looking at big data like this, there are millions of possible interactions that we're looking for between all of these biomarkers. Because there are millions of biomarkers that we're looking at, and there's so many possible connections out there. And generally, in the field, if your hit rate is above 50%, it's considered really good. Because when it comes to big data, these are really hard problems, and so 16 out of 18 we were thrilled by, right? Like we were expecting around 50%. And my scientific collaborators say that even if only 20% of what we identify is useful, that still saves them a huge amount of time and money. So yes, these are hard problems. And again, these are not the end answer. These are just predictions that then people go back and test. Yeah. Yes.
So how many sources do you use for your big data? Like for your microRNAs, how many databases are you mining?
Yeah, so the database that we're using for this project was the Cancer Genome Atlas, and this was actually compiled by the National Institutes of Health. And this is the most comprehensive database of genetics for cancer that's out there. What we typically do is we build our models using one database, and sometimes we test them on another. We do this especially when we don't have collaborators that can test our predictions in the lab. Then we use other databases to do this. But people have actually already gone through and actually combined and compiled a lot of this data into databases. That doesn't mean the data is easy to access or easy to use. Actually, some of the software we developed was actually just how on earth do you even access this data and merge this so that you can even analyze it? It's not trivial, right? Like we have to do a lot of kind of computer engineering to make that happen. But there's a lot of data out there. Yeah. Yes.
So at the beginning, you talked about how we're at that 90% of the data has been kind of captured in the last 2 years, and we're headed towards even greater explosion. So going forward, what are the challenges to managing that data? Do we have hardware problems? Do we have theoretical problems, like we don't understand how to manage that much data? Or what do you see as the challenges of addressing?
Yeah, so it's a great question. The question is really about data management. So we've got tons of data, and how do we manage all of this data? So it is a huge challenge. There are major hardware challenges that are associated with that, and also just even accessing the data to analyze. Some of these data sets are so big that we cannot load data into memory on our machines, right? I have really big server systems and that have tons of RAM, and we still can't load all of the data at once into RAM. So we are developing this is at the interface of computer science and statistics and applied math. We have to develop algorithms that can only see parts of the data and somehow still find the solution and still fit a joint model when you can only access parts of the data. And this is really important because this is how as the data gets bigger and bigger and bigger, we're never going to be able to access all the data and load all the data, and we have to develop algorithms like this. So that's just one example, but it's a huge challenge dealing with the sheer size of the data. Yeah. Yeah.
There's a federal law that protects patients' health information. So how does all this data get out into an accessible form?
Yeah, so this data is considered de-identified human data. You do have to sign a data agreement to get licensed to access this data. This data isn't not just anybody can go and access this data. But this type of data is difficult from gene expression and epigenetics to reconstruct who that person is. And the type of data that would allow you to identify an individual would be whole genome sequencing, and that data is never made publicly available for sites like this. So what we get instead is a very small snapshot of mutations in those tumor cells, and those are specific to just those tumors and not specific to that person's DNA, so they're considered non-identifiable data. But in general, this is an issue. This is a major issue with electronic health record data. where you do have personally identifiable information, and there are a lot of hurdles for data scientists like me to access that type of data because of it. Yeah. Okay, one last question. Yep.
Would it be possible that in your data, the answer to some of the excellent questions that Bob Curl brought up in the previous talk may lie the answer?
I maybe didn't quite understand. Wait.
Well, we got all this data.
Yes.
At the same time, we've got all these problems and questions. Could one help the other?
One, I'm not sure that I quite know how to answer that, but that could be very
I don't
interesting.
either.
I could talk to you.
I don't even understand my own question.
Perhaps I could talk to you afterwards about that. Anything else? Okay, I think we're about done. Okay, thank you very much again.